So the next speaker here, we have lovely Janet Tate. Janet is a developmental biologist, and she's originally from Canada. She's currently completing her PhD at the Biomedical Centre in Munich at the LMU. And before that, she was actually in Scotland completing her master's degree. So Janet works closely with the African clawed frog, and today she's going to guide you through the very intricate processes within development. Welcome.
All right, thank you so much for the introduction. Yep, my name is Janet, and I'm going to be talking to you about my research as a PhD student in developmental biology. So, over the course of embryonic development, an animal goes from a single cell, a fertilized egg, to this coordinated, complicated bundle of trillions of cells that we call a human, or a cat, or a frog. And not all of these cells are alike. In fact, there are hundreds of distinct cell types in the body, but they all contain roughly the same DNA. So if that's true, and underlying this huge diversity of cells is the same genetic blueprint, then how do we end up with these completely different cells, like cells in your eye that help you see, cells in your gut that help you digest, cells in your brain that help you think? Well, that's a really good question, and something I'm totally fascinated by. And it largely comes down to which genes in the cell are turned on or off at which time during embryonic development. So, if you were to take all of the DNA contained in one cell and stretch it out end to end, you would get a thread that is 2 meters long. Yep, still going. All right. See, it's taller than me. And this needs to be compacted into a cell that's not even visible to the naked eye. So that's basically the equivalent of me hucking this thread out my car window, driving 20 kilometers, and then trying to fit all of that thread into a ping pong ball. Sounds impossible, right? And what's even more astounding is that specific molecules need to be able to get to specific pieces of this thread at very particular times in order for development to progress normally. So how do they accomplish that? How do we get these very specific molecules where they need to be at the right time? Well, unlike the thread that I've got here, the thread of our DNA is not so jumbled. So the same way that I would wind my thread around a spool like I'm doing now, we wind the thread of our DNA around a molecular spool that's known as a histone. And these histones can be modified in certain ways to change the way that the thread winds around the spool and influences which genes are turned on and turned off. And this is done by modifying histone tails. And you can think of a histone tail kind of like a barcode. And the different molecules that come in and interact and change this barcode are like this. So we've got writer molecules that can come in, add new lines to this barcode. And then we've got eraser molecules that can come in, get rid of those lines, change it up. And then we've got reader molecules that come in like these tiny molecular scanners. They read the information that's on the barcode and spread that information around. And the way I study this, I think, is pretty cool. So I work with this frog, a very funny-looking frog called Xenopus. And Xenopus is Greek and Latin for strange foot. But I would argue that the feet are actually one of the least strange things about these frogs. They have a really rich history of use in science. So, for example, they were used in some of the first cloning experiments. They were actually one of the first fairly accurate pregnancy tests. And they've even been to outer space. So what I do on a sort of weekly basis is I take eggs from these frogs, I fertilize them, and then with the tiny glass needle under a microscope, I inject our histone writers, our barcode writers, and our barcode erasers, and let these frogs grow up a little bit and try to understand how modifying our histone barcode modifies embryonic development. And it's my goal as a PhD student and aspiring scientist, I guess, to contribute to this knowledge of this completely complex but completely universal process of embryonic development. So thanks so much for listening, and I'd be happy to take your questions.
Wow, so cool. Thank you very much. everything you talked about occurs at such a tiny, minuscule,
Yeah.
scale. It's almost difficult to contemplate with your mind. So anyways, enough of what I think. What do you guys think at home? Get your questions coming in. Don't be shy. I've heard developmental biologists don't bite. And we've already got some coming in now. So the first one is, what can we learn about your your research, from your research about human development?
Okay, that's a really good question. So despite the fact that frogs and humans look so completely different, we're actually quite the same on a kind of molecular basis and on how we develop. So if you think about it like music, so you've got classical music that sounds like one way, and then you've got like rock or pop or something like that. The genres sound so different that it can be hard to see, to kind of hear the similarities about it. But if you start to dissect it, you'll hear, some of the same instruments are being used, some of the same kinds of notes are being played, and some of the same themes and this sort of thing. It's actually quite similar with how we develop. So humans and frogs, despite the fact that the end product looks so different, they're actually made up of a lot of, so they contain a lot of the same genes, a lot of the same biological processes are used, and at a kind of molecular level, the building blocks of a human and a frog are not so different. So there's actually a lot we can kind of learn and gain about humans from studying development in frogs, because it's really not so different. It's just these end products that look so different.
Huh. So that might answer the next question, which we've actually got from Anne on YouTube, and Anne is actually from Toronto. And she's asking what similarities exist between frog and human development. Mm-hmm. So I don't know if you sort of touched on that in your last answer, but maybe you want to add to that.
Sure, yeah. So again, the sort of underlying processes are similar in a lot of ways, but they branch off into these very different looking and functioning animals. So, I think part of this is because we're actually quite related on an evolutionary level to frogs. Since we're both vertebrates, we share a common ancestor that's actually not so far in the past. Okay. Compared to, say, like, bugs and humans are much less related, or plants and humans and this sort of thing. So, a lot of the kind of, things are shared, but obviously resulting in very different end products.
Okay. So another one from Luke on YouTube. He's asking, you said the frog was one of the first pregnancy tests. How on earth did that work? I mean, that's a very good question, Luke. I agree.
Yeah, it's a pretty funny story. I mean, these frogs are from South Africa. And at the time, there was a scientist who was researching hormones. Mm-hmm. And he found that if he injected an extract from the pituitary gland into the frog, the next day it would lay eggs. And so this was really useful in the lab because, of course, if we're doing experiments on frogs and development, we need a lot of eggs to be able to work on them. So this way he could induce the frogs to lay eggs whenever he needed them. Mm-hmm. But then he started to think, like, are the hormones that are making the frogs lay eggs kind of similar to what we can find in humans? And he found that by injecting the urine of potentially pregnant women into the frog, if they actually, the urine,
oh, wow.
It's like peeing on a stick, but a little more.
It literally is,
right?
A
little more dramatic than that. Yeah. So, so basically, if the woman was pregnant, the next day the frog would lay eggs. If not, then no. And this was like staggeringly accurate, like very few false positives and very few missed pregnancies. So, yeah.
That's amazing.
Yeah.
Who'd have thought that? Mm-hmm. Another question from Ash on YouTube. When you modify the histones, what aspect of limb development are you interested in, or are you looking at?
So I don't actually look at limb development, but I mean, there are lots of really interesting questions you can answer by studying limb development. I actually am interested in a very specific cell type called multiciliated cells. And these cells show up on the embryos of the frogs after about 2 days of development, and they're actually not really there for that long. So there are these cells that are in the skin, and they basically beat liquid down the kind of side of the embryo, and it helps mostly to spread antimicrobial like agents down the skin. And they're actually quite similar cells to what we find in a lot of different organs that need to generate fluid flow. So, for example, the these kinds of cells are found in our lungs. They help to clear mucus out of the lung. They're found in the brain, in the intestinal tract, and this sort of thing. So by using these frogs, since the embryos develop quite quickly and develop sort of outside of the mother, it's a really great system to study these cells. And yeah, so
Great.
yeah. No limb development, but
But still, super interesting. So you mentioned again about the histones with the red pen, right?
Mm-hmm.
So Mart is asking on YouTube, do you know how many histones there are in the human body?
That's a really good question, and I have to admit that I have no idea. Lots. I'll leave it at that.
Okay. And another one from two users asked the same question.
Okay.
Are there any known diseases in humans that are due to faulty histones?
I'm sure that there are,
Yeah.
but off the top of my head, nothing's coming to mind. But yeah, I mean, histone modifications, histones and histone modifications are really essential to how we function and how our DNA manages to kind of regulate itself and compact itself. But yeah, off the top of my head, I'm not being able to think of anything.
Okay. And I also just wanted to mention that Ash, who asked that question, is actually from South Africa, like the frog that you work with. So hi to Ash.
Ash, have you ever seen them out there?
Yeah. Another question here is, for some reason, a user here thought that embryonic development is extremely well understood, and it was already sort of well known, particularly in frogs. So what really what really needs to have more research put towards it? So what more do we need to find out?
Yeah, it's a really good question as well. So yeah, while, I mean, embryonic or developmental biology as a field has been around for a really, really long time, and a lot of studies have been done kind of morphologically and this sort of thing, watching an embryo, how it grows and kind of detailing the steps. But as science progresses, as our tools get better and better for looking at certain processes, then it opens up questions that we wouldn't have ever kind of thought possible. So now, being able to do more kind of whole genome studies or proteomic studies and this sort of thing, we can learn so much more about developmental biology. And yeah, biology in general, it's so complicated. There are always more questions that we're not going to be able to answer. So yeah, while a lot is known, there's always more to understand.
So when you're working in the lab, there's another question from one of the users, Luke, on YouTube. He's interested in what type of sort of equipment do you use to study the frog development?
Yeah, so on a kind of basic level, every week or whenever I need the frogs, I'll take the frog eggs and I basically pull these tiny little needles, and I can use them to inject different kind of reagents, let's say, that can block genes or overexpress, turn on these genes in different ways, and then even just by simply like looking under a microscope, I can see what changes have occurred. But we do have a lot more kind of sophisticated techniques that we can use to study these frogs. So from kind of very fancy microscopes to so like sequencing technology, like we can look at how many genes are turned on and to what extent at different stages of development or with different kind of experimental conditions put onto them. So we have a lot of really cool tools we can use to study this.
Great. And obviously the frog itself is sort of a tool, right? Because it's modeling human development. So another user asked Eva, what are the advantages of using frogs over other model organisms, such as the mice or other animals?
Yeah, so a really big advantage of the frogs over mice is that while mice are mammals and they develop inside of the mother, the frogs, they lay their eggs and then they can grow up just kind of in the water. So they're really easy to look at, to image, and to watch their development. So that's one really good benefit. As well, these frogs, as the South African researcher figured out, if you inject a certain hormone into them, they'll lay eggs anytime. So that makes it really easy to kind of collect material and, to get enough to study. In addition, the embryos are transparent, so it makes it really easy to take pictures of them and, on the inside and this sort of thing. So, and their eggs, I mean, okay, if we're thinking chicken eggs, they're not that big, but for frogs, for kind of embryos we can study in the lab, they're actually about the size of a poppy seed, the eggs. So that doesn't seem big, but it's actually big enough to allow a lot of like really interesting kind of manipulations and under the microscope. So yeah, makes them pretty good and widely used model organisms.
Great. So another fun question which I really like is, did you know if frogs can have identical twins? Have you ever known? I mean, I don't know if you look at a frog and you recognize them, you're like, oh, that's Stanley or something, and this is his identical twin. Is it possible? Because that would be very interesting within the developmental question.
Right. So it's kind of hard to say because, well, I would say identical twins as we think of them in humans, it's usually, so, you know, there's an egg that it ends up kind of giving rise to two different twins from the same starting sort of embryo.
Okay.
Sorry, I'm kind of butchering that explanation, but no, I don't think frogs really end up having identical twins, partially because actually we fertilize them in the lab.
Okay.
So it's already separate, distinct eggs that we're fertilizing, and yeah, I don't think they.
So it's impossible to simulate this. What happens when we have identical twins in the lab?
I don't really know.
Okay.
Maybe it is, but yeah, I haven't really thought about that.
Okay.
So, interesting question, though. I'll definitely follow up on that.
Another one from Mar on YouTube. Does the mom's health affect epigenetic development?
Yeah, so that's actually a really exciting aspect of studying epigenetics, which is kind of these changes to gene expression without actually affecting the DNA. So basically, like with our histones, the thread that I was winding around the spool, that stays the same. It's just this barcode that changes, is that there can be these heritable changes that don't result from changes in the genes. So external kind of stressors or conditions can change how the offspring develops. So that's, very cool.
Okay, so just back to basics. Like, what is epigenetics, actually, for those who don't know at home?
Yeah, so there are kind of a few different definitions of epigenetics. One is these heritable changes that aren't due to a change in DNA. But in a more broad sense, people tend to think of epigenetics as sort of changes of gene expression that are due to maybe changing your histones or changing or modifying DNA after it's been formed. So basically, it ends up being these changes to your gene expression and to how you develop or, without actually changing the DNA. Sometimes it's heritable. That's kind of the classic definition, but sometimes no.
Okay, so let's get back to a simpler question. How do you get the frogs? Do you go and hunt them and collect them and bring them back to the lab, or how do you do that?
So I have met researchers that do that. They go out and they bring back colonies and this sort of thing. Yeah, I mean,
even in Munich?
Not in Munich. But basically, because they're such a widely used model organism, there are a lot of kind of resources and places that will, culture the frogs, grow them up, and then sell them to labs that want to work with them.
Oh, okay.
So we basically order them from a frog growing facility, I guess.
Okay.
Yeah.
Great. And another question is there any way that we can know the change our histone, or is the process only biological? So can we physically see the changes our histones makes, or is it only biological so we don't see? So if your histones change something to your DNA, do you see that on your appearance or something like this?
So yeah, it's really interesting. I mean, you're not going to actually be able to say, okay, I'm going to look and see that this histone has changed, but because histone, the condition of our histones affects which genes are being expressed and which are not, you can see changes that result in changing the histones. Like, for example, when I work in the lab, I basically really mess with this one specific barcode line, you could say, and you really do see it in the embryos. So these cells that I was talking about before that are on the embryonic skin, we completely lose them. So, I mean, it is a biological thing in the sense that, you're not going to be able to look at your own skin and say, oh, my histones have changed. But you can see the effects of that are caused by changes to your gene expression.
Okay, so we can't see very obviously the results of histone changes. But another question here on YouTube is, can we change our histone modifications by, for example, foods that we eat or our sun exposure or environmental factors?
Yeah, so that's what's really exciting about kind of these heritable changes, is that external factors can actually affect our histones and what's going on. And then in some cases, these modifications can be passed down. So, yeah.
So we can so the mother can pass them down to the babies?
Yeah.
Just because the mom ate something different?
Yeah, so there actually have been a lot of studies done on obesity and this sort of thing, and how it can pass on cardiovascular problems to
no way.
So there's a lot, it's a lot going on. Your histones are really important and exciting.
Well, I think we're sort of running out of time now, so I'd like to thank you very much for your time, and have a nice rest of your evening.
Great, thanks so much.
So once again, if you didn't manage to get your question answered tonight, don't worry, Janet is taking questions as well via her Instagram and her Twitter account, which we'll post in the feed right now.